Hi, hello, and welcome to my channel, my YouTube channel. Please make yourself comfortable, make yourself at home, please. And my name remains Amira. So, on my channel, I teach. So, today I'm going to be teaching on molecular cloning. So, please sit tight and enjoy the ride. So before we go into molecular cloning, we have to first understand what cloning is, because molecular cloning is basically a subset, a division of cloning. So first off, we have to understand what cloning is, just a brief definition. Now, cloning is simply the process whereby organisms with identical or virtually identical DNA are created or produced. So it means you have, for example, one person, one organism, one thing, and then you want to make something else that's extremely identical, if not the same as the first one. So basically, they have the same DNA. Remember what DNA is. DNA is that segment, that part of your nucleus. You know, the nucleus is found in the cell. So DNA is found in the nucleus. And it's the DNA that carries genetic materials that is responsible for the way I am, the way you are. So, and the DNA is literally the storehouse for genetic materials for creating every single part of each organism. So now, in cloning, you make organisms that have the same DNA, or virtually the same DNA. Now that's generally what cloning is. Now move on to molecular cloning, now that we understand what cloning is all about. So like now you have a baseline understanding of what cloning is. If you see it in all these sci-fi movies, you'd be able to tell, oh, I actually do know what cloning is, and more than just what I'm watching. So like you'd have that personal intel on what's going on. On to molecular cloning. So since we know what cloning is, molecular cloning is one of the branches or subcategories of cloning. Now, molecular cloning simply involves um, artificial the artificial process used to amplify DNA segments. So, like in molecular cloning, you do not clone the full DNA, you literally just take segments of the DNA, which basically is a gene. So you literally just take a gene segment, a part of the DNA, and then you clone just that part. You amplify it, which means you make it more, you express it to be more. So in molecular cloning, you're actually making a segment of gene, making a very similar segment of that gene. So you get the original one and then use molecular cloning to make many other copies of that same segment, so they look exactly the same. They actually are exactly the same. So that's basically what molecular cloning is. So for molecular cloning, there are actually a series of steps involved. Now, there are 4 steps for molecular cloning, and these steps are: number 1, fragmentation; number 2, ligation; number 3, transection or transformation; and then number 4, we have screening or selection. Those are the steps in molecular cloning. Now, for step 1, where we have fragmentation, the specific DNA segment of interest is simply coded out using restriction enzymes. Now, in DNA or molecular cloning, or also you can call it gene cloning, segment is double coiled structure, this double helix, and then it has bases, nitrogenous bases. So you have adenine to, you have guanine to cytosine, adenine to thymine. So guanine to cytosine, then thymine to adenine. So you have DNA segments. You have the full DNA and then you have DNA segments. Now, for molecular cloning, we are making more copies of specific segments of the DNA. For example, we might want to take out these segments, this AGCT, and then make more copies of it because that's our gene of interest. So this would be our gene of interest. Now, to isolate our gene of interest, to bring it out from here, we'd have to use certain substances, certain enzymes. They are called restriction enzymes, RE. So now, the restriction enzymes would go to the DNA segment and then cut and cut. And then we will have that DNA segment, that particular gene of interest, set free. So we would use the restriction enzyme to cut that specific segment of DNA to get the gene. Now, restriction enzymes they have specificity. They are very specific for the particular segment of DNA which they act on. So you'd see a particular restriction enzyme would only act or would only cut specific regions of the DNA. They would only cut specific regions. So you'd have to check for the restriction enzyme that would cut that particular segment of DNA you're interested in working on. So basically, in fragmentation, we cut out the gene of interest from the DNA, for example, AGCT. Then we also have to cut out a certain segment from the plasmid vector. Remember, we're getting this plasmid from the bacteria. So just get, for example, this is the bacteria. This is the normal nucleus of the bacteria, and then this is plasmid. So the plasmid is a circular material, a circular nuclear material that is separate from the nuclear material that controls the bacteria. So we isolate this plasmid, bring it out, and then using the same restriction enzyme, we cut out a portion of the plasmid. So that restriction enzyme works here, cuts out this portion, and then the plasmid would have a missing portion. Now this missing portion, our gene of interest would fit right into this position because the restriction enzyme has cut out a part of the plasmid where the gene of interest would be able to fit right into. So that's basically what happens in fragmentation, the first step of molecular cloning. Is ligation. So in ligation, this simply is a step whereby an enzyme ligase joins that plasmid together with the gene of interest. We've already cut them out in fragmentation. So in ligation, the enzyme ligase simply forms new phosphodiester bonds between the plasmid and gene of interest. So basically, DNA ligase comes in and forms phosphodiester bonds. So it thus brings the plasmid and our gene of interest together to form a complete plasmid. Now, this structure formed at this point in time from the fusion of the plasmid and gene of interest is called the rDNA, full meaning recombinant DNA. So this structure formed is rDNA. And this brings us to the end of ligation. So ligation just means the enzyme ligase joins the plasmid plus gene of interest to form the recombinant DNA. So that's basically the end of ligation. So next step is the third one, which is transformation. Now in transformation, remember the bacteria, the one we got the plasmid from, that circular DNA material. So we get back the bacteria and then we put the recombinant DNA, this one we formed here, we then insert it back into the bacteria. Now, when we insert this recombinant DNA into the bacteria, it makes it become transformed. So that's basically the process of transformation. Yes, so putting the recombinant DNA, remember the recombinant DNA is literally the plasmid plus the gene of interest. So putting it back into that bacteria cell, which we call the host here, makes it transform. So that's the process of transformation. That's transformation. But then it's not, so direct for transformation. For the bacteria, the bacteria cell has to be made competent enough to take up the recombinant DNA, and this could take a series of processes. You can use cold calcium chloride, you can use electroporation. These are just a series of steps to make the bacteria be able to take up the recombinant DNA. And then once that has happened, that's basically transformation. Now this brings us to the fourth and final step in molecular cloning, which is screening or selection. For selection or screening, just like the name suggests, what we do here is to screen or select the particular bacterial cells that were successfully transformed, the particular bacterial cells that successfully took up the recombinant DNA. So at least this way we can be sure that, these particular bacterial cells contain our gene of interest, because basically the whole reason we've been doing all of this is just so the bacteria can clone our gene of interest. That's what we're interested in. So we have to, this is just like a step to check, to confirm if the bacterial cell actually successfully took up the recombinant DNA. And how does this happen? There are a series of processes to do screening. You can use you can use specific selection markers, but the one I'm going to explain is using an antibiotic, using antibiotics. So antibiotics, they work against bacterial cells, so they can kill these bacterial cells. So what we do here is that we select a specific antibiotic that our gene of interest is resistant to. That is, if you should apply that antibiotic to a bacterial cell that has our gene of interest would help the bacterial cell to resist that antibiotic and thus help it to still survive in the presence of that antibiotic. But if that bacterial cell does not contain our gene of interest, it won't have anything to help it resist the antibiotic, so it would die. So that's basically how this selection happens. So we just um, choose a specific antibiotic based on the kind of antibiotic resistance gene our gene of interest is, then apply it to the bacterial cells. So it would kill off the ones that haven't been transformed successfully and leave only the ones that contain our gene of interest. Now that's how selection or screening is done. After the selection or screening, these bacterial cells that have been confirmed to be transformed will then continue to grow. They will be put in an environment that supports their growth, that supports their fission, their cloning, which an example of such an environment is a fermenter. So the bacteria are grown in a fermenter where they grow and form colonies. Now, this is basically the process of molecular cloning. So, and molecular cloning has so much importance in therapeutics, genetic engineering, so many mind-blowing fields that are making advances in health, medical sciences, and all of that. So, you see, molecular cloning helps in production of drugs. You know, the commercial insulin that is marketed that helps diabetes patients manage their conditions. It's via molecular cloning that you get this insulin. And it's not only insulin, there are so many other genes that are helping so many medical conditions, genetic engineering, all of this. Molecular cloning really has vital importance in all of this. So, this brings us to the end of this video. Actually, it has been so much fun, this entire process. I hope you gained something. I hope, I don't know, molecular cloning, the whole thing is now very, very clear. This is the first video on molecular cloning. I would still make another video that would show specificity, that would be very specific with examples, and with examples therapeutic settings where molecular cloning is used to produce drugs to help people with so many health issues. So thank you so very much for your time. Thank you so very much for watching the video. I hope you enjoyed it as much as I did. So my name remains Amira. I hope to see you next time in the next video. Thank you very much for watching.